Good morning, children. This is the H part of the structure and germination of seed. And in the previous video, children, I have explained you about the type of germination, that is the apigeal germination. And in this session, I am going to explain you about the hypogeal germination. When the cotyledons remain under the soil or just on the ground during germination of seed, it is called hypogeal germination. So children, in hypogeal germination, the cotyledons remain under the soil. And in apigeal germination, you have seen that the cotyledons come above the soil. And why these cotyledons remain under the soil in hypogeal germination? It is because of the rapid growth of epicotyle that the plumule emerges out of the soil. Now, why these cotyledons remain under the soil? This is because of the rapid growth of the epicotyle. And because of that rapid growth of the epicotyle, the plumule emerges out of the soil. It is found in the seeds of gram, pea, maize, mango, groundnut. And these are the examples where you can see hypogeal germination. So various stages during hypogeal germination of maize, that is a monocotyledonous endospermic seed. As you know, children, this maize is a monocot seed and it is endospermic. Endospermic means the food is stored in the endosperm. And pea, it is a dicotyledon seed and it is non-endospermic. That means the food is stored in the cotyledons in this. So here today we will study about the hypogeal germination in maize, and the biological name of maize is Zea mays. The grain absorbs water and swells. The cotyledon absorbs food materials stored in the endosperm. As you know, children, when we sow the seeds in the ground, all the seeds, they absorbs water and the seed coat swells. In the same way here also, the grain absorbs the water and swells, and the cotyledons, they absorbs the food materials which are stored in the endosperm because it is an endospermic seed. The radicle peers through the protective root sheath, that is coleorhiza. See children, we have seen that this maize or in monocot, there is a protective coat, or we can see a protective sheet above the root or the radicle, and that sheet is known as the coleorhiza. So this radicle, it peers out through the coleorhiza and it grows downward into the soil. The radicle develops into primary root with the root hairs. After that, this radicle peers out through the root coat, it grows down and it develops the primary roots with root hairs. The coleoptile elongates and peers out of the grain in the form of a tube opposite in the direction to the coleorhiza. Now, as the radicle is protected by a sheet, that is coleorhiza, in the same way, the plumule is also covered by a protective sheet, and that sheet is known as the coleoptile. So, the coleoptile elongates and peers out of the grain. Now this coleoptile, it peers out of the grain in the form of a tube in an opposite direction to the coleorhiza. As you know, children, the radicle grows downward and this plumule grows upward. So both of them, they are in the opposite direction. You can see here, children, this is a maize grain and radicle is coming out first, piercing out through the coleorhiza, and this coleoptile emerges out in the opposite direction to the coleorhiza. Then here you can see the root and root hairs also. A cluster of fibrous root arise around the base of the primary root. You can see here, this is the primary root and the cluster of roots appear here. The first leaf of the plumule peers out of the coleoptile, which now forms a tube-like collar around its base. See, these are the first leaves. This is the plumule, and now it arises. The first leaf of the plumule peers out of the coleoptile, which forms a collar around its base. Around its base, here you can see a collar like, and around its base. Now more leaves appear in succession. Primary root perishes, giving rise to a fibrous root system. Now after some times, when the first leaves appear, the primary root, this is the primary root, it disappears, and what happens, this it perishes, we can say, and it give rise to a fibrous root system. Now this is all about the hypogeal germination in maize. Now we will see the hypogeal germination in the seed of pea, that is Pisum sativum. Pea is a dicot seed. Now the seed absorbs water and swells. Food reserves in the cotyledons become soluble by the action of enzymes and pass in the tips of the growing plumule and radicle. So here also the seed absorbs water, the seed coat swells, and what happens, the after that absorbing water, the food reserves in the this cotyledons, it becomes soluble by the action of the enzymes, and it passes to the tip of the plumule and the radicle. Now the seed coat ruptures after absorbing water, the radicle elongates, emerges out of the unruptured seed emerges out from the ruptured seed coat and grows downward into the soil. Now what happens, the seed coat ruptures and the radicle elongates from the ruptured seed coat, and it grows downward into the soil. Now the epicotyle begins to elongate and forms a small loop. Now the epicotyle begins to elongate and forms a small loop. You can see here, this is the here this is at the epicotyle. You can see here this loop like this one. You can see here this loop, it forms a loop like structure. Root hair appears to in the region of the radicle where elongation has ceased. Now you can see the root hairs, and it appears in the region where the we can see elongation has ceased. Now the epicotyle elongates rapidly. The plumule located at its tip becomes free from the cotyledons by the straightening of the epicotyle loop. Now what happens, see children, the epicotyle, here you can see the epicotyle, it elongates rapidly. The plumule is located at the tip of the plumule, which is located here at the tip, becomes free from the cotyledons. These are the cotyledons, free from the cotyledons by the straightening of the epicotyle. You can see here the epicotyle becomes straight. Here it is in the form of a loop, then it becomes straight. Now young leaves appear and prepare food for the plant. Seed coat and cotyledons remain below the soil. Primary root persist, give rise to a taproot system. Now here what happens, see children, the young leaves starts appearing, and the seed coats and the cotyledons, they remain below the soil, and the primary root system here, this primary root persist, and it give rise to a taproot system. So this is all about the hypogeal germination in a dicot seed. Now the difference you can see between the epigeal germination and hypogeal germination. In epigeal germination, children, the cotyledons come above the soil, and in hypogeal germination the cotyledons remain below the soil. And in epigeal germination, children, hypocotyle elongates, and in hypogeal germination the epicotyle elongates. So children, this is very important. Learn the difference between hypogeal and epigeal germination. Now a very special type of germination is there, that is the vivipary. Another type of germination, that is vivipary. Now what is vivipary? We will see here. The seed of the plants growing along the seashore or in saline marshes show a special type of germination called viviparies. So children, here the seeds of the plants which grow near the seashore or in saline marshes show a very special type of germination. So what is vivipary? In this type of germination, the seed germinates inside the fruit while it is still attached to the parent plant. Till now we have seen the seeds are dispersed, then the seeds are sown. We can see the fruits become dry, and we collect the seeds and then we sow the seeds and then the germination starts. But in this type of germination, children, the germination starts, seed germinates inside the fruit and that fruit is still attached to the parent plant. A club shaped radical emerges out of the fruit. You can see here a club shaped radical. This is the club shaped radical. It emerges out of the fruit. This is the fruit. You can see. And the germination has started. This is the club shaped radical. The lower part of the radical gets swollen. You can see here the radical gets swollen. Finally, the seedling gets detached from the parent plant and gets embedded in the muddy soil below. Now, after this, when this radical becomes swollen, what happens? The seedling gets attached from the parent plants and from the parent plant, and the lower part of the radical, this lower part of the radical, this embedded in the muddy soil which is below. The radical develops this lateral branches quickly to anchor the seedling. You can see this branches appearing in the radical so that it may anchor the seedling into the soil, that muddy soil. So this is about the vivipary. So the seeds which grow starts germinating inside the fruit while it is still attached to the parent plant is known as vivipary. And this type of germination is found in Rhizophora, Caryopsis and Senorita. These are the examples of viviparis. So children, here we end this chapter of the structure and germination of seed. I will send you the assignment. Do the assignment in copy. Bye.